Hi everyone, welcome to my channel Pure Biology, and today's topic is that N-linked glycosylation of protein. Okay, this N-linked glycosylation of protein is one function of the endoplasmic reticulum. Okay, and before in previous video I have already told that the actual functions of the endoplasmic reticulum very briefly, and in next videos I will try to explain their function properly. Okay, so you just support me for doing the best, and obviously support my channel to run it properly. Okay, so let's continue that the N-linked glycosylation of the protein. What is called this glycosylation? This is the attachment of the sugar molecule to the nitrogen atom of an amino acid residues of protein. You all know that the simple form of the proteins, these are the amino acids. So glycosylation meaning is that the attachment of the sugar molecule with the N atom of the amino acid. And here, and this is possible in RER, means you all know the two types of endoplasmic reticulum. One is the rough endoplasmic reticulum, another is the smooth endoplasmic reticulum. So this is the actual function of the RER. So this total process is occurring in the RER. This in RER, this process, that the involvement of some substances are there. One is the oligosaccharide precursors. Okay, precursors means not mature oligosaccharide, because after biosynthesis or synthesis of the oligosaccharides, the mature oligosaccharides are formed. And next one is that the dolichol. Okay, so what is saying that in RER the process involves the addition of a large performed oligosaccharide precursor to a protein, and this to a protein. Okay, so here the oligosaccharide precursor is very important for involving this glycosylation process. And what is happening here? The precursor, that means this oligosaccharide, is linked by the phosphoryl residue. Okay, oligosaccharide is connected with a phosphoryl residue, and obviously these are all connected to the dolichol, and this dolichol is very long chain of the polyisoprenoid lipid, and it acts here just like a carrier, or you can say that it helps in the transfer of substances. Okay, and here what is happening, that is firmly embedded. This dolichol is embedded in the RER membrane and act as carrier. Okay, and this oligosaccharide biosynthesis, what is happening in the first situation, or how it begins? This begins on the cytosolic phase, okay, of the endoplasmic reticulum membrane. This is happening in the cytosolic phase, and with the transfer of N-acetyl glucosamine to dolichol phosphate. So first of all, this N-acetyl glucosamine, this transfer to the dolichol phosphate. Clear? And that's way the biosynthesis of the oligosaccharide that begins. Okay. So next situation, the two glycosamine and 5 mannose residues generally added to the dolichol phosphate. Okay. So here first the transfer of the N-acetyl glucosamine to dolichol phosphate, and generally 2 glucosamine and 5 mannose generally attached with the dolichol phosphate. Okay, and obviously these occur in the cytosolic phase. The first sugar and the N-acetyl glucosamine, first sugar means glucose and N-acetyl glucosamine, these link to the dolichol by the phosphate bridge. Okay, that means phosphate, with the help of the phosphate bond, the first sugar and N-acetyl glucosamine both attached with the dolichol. Okay, so next is that this high energy bond activates the oligosaccharide for its transfer from dolichol to aspargine. That means due to the formation of this phosphate bridge or phosphate bond, what is happening? The transfer of the oligosaccharide is possible from follicle to the aspergine, and this reaction is possible one due to the presence of one enzyme, that is the transferase. Here is the oligosaccharide transferase. This enzyme has important role during the transfer of oligosaccharide from follicle to aspergine. Okay. Next thing is that the 7 residue dolichal phosphoryl intermediate is flipped to the luminal phase. Okay, and this transfer is possible in this aspergine, where this is the side chain of the nascent polypeptide. This aspergine is the side chain of the nascent polypeptide, or you can say that the transfer of oligosaccharide from the dolichal to the aspergine, this is the nascent polypeptide chain, part of or side chain of nascent polypeptide chain. Okay, and where this is present? This is present in the luminal side of the area, not the cytosolic side. This is the luminal side of the area where this aspergine is present. Clear? Next is that after 7 residue dolichal phosphoryl intermediate is flipped to the luminal phase, the remaining, what is saying that? The remaining 4 mannose, all 3 glucose residues are added one at a time in luminal site. Okay, so here it is saying that 7 residues of this dolichal phosphoryl intermediate that flip to the luminal phase, they are already 4 mannose and 3 glucose residues added just side of the luminal phase. Okay, and the sugar molecule, what is the actual function of the sugar molecule, or how the sugar molecule generally help in the oligosaccharide synthesis? Generally, first they are activated in the cytosol and they form the nucleotide sugar. Okay, because actually the nucleotide sugar is very important for this, not the same simple sugar. Nucleotide, first they convert them to the nucleotide sugar and then they donate their sugar to the lipid. Okay, first they convert them into the nucleotide sugar and then after that they donate this to the lipid, but in the sequence order. Clear? And after that 1 mannose and 3 glucose residues are removed by the enzymes. Okay, as the transfer is totally completed, then 3 glucose residues and 1 mannose that are removed by another enzymes. So this is the end-linked glycosylation or glycosylation. And this end-linked glycosylation is very important. Why? Because there are some functions of the that is some role of the this glycosylation, or you can say that oligosaccharide, because some glycoproteins are there that are helping in cell adhesion. Okay, and this is only possible due to the presence of the mature oligosaccharides. Okay, another thing is that some glycoproteins, these are actually some as antigen, but without oligosaccharides these glycoproteins cannot act as a antigen. Okay, and third point is that end-linked oligosaccharides confer stability on many secreted glycoproteins. Okay, that means it gives also stability to some glycoproteins. So if oligosaccharides not mature or it remain in the precursor level, so in that situation the glycoproteins not gain the stability. So that's why the oligosaccharide biosynthesis is necessary, and this is occurring in the endoplasmic reticulum, obviously RER. Okay, this is the first function of the endoplasmic reticulum. Here I tested, and you can consult any book just after seeing this video. Okay.